Welcome to the Metabolic Classroom. I'm Professor Benjamin Bikman, a biomedical scientist and professor of cell biology. Today, we're diving into a molecule that most people associate with liver disease and jaundice, but it actually has a fascinating and underappreciated role in metabolism. The molecule of interest today, the focus of the lecture, is bilirubin. You have heard of bilirubin, I bet, to some degree or in some way. It is a yellow pigmented molecule. In fact, that's kind of what's made it famous for reasons we'll get into in a moment. But it comes from the breakdown of hemoglobin in red blood cells. When a red blood cell has reached the end of its lifespan, usually about 120 days, it's broken down in the spleen. And then the heme portion, so when you think about the red blood cell, there's a part of it called hemoglobin. Well, that consists of two parts, and one of them is heme. When the heme portion is broken down in the process of getting rid of the dead red blood cell, it's converted into a molecule called biliverdin, which is then further converted into the famous or infamous bilirubin. This process is part of the body's way of recycling iron. So getting the iron from the blood cell and then trying to reuse some parts of the components of the red blood cell. Now, once bilirubin is produced, it is released into the bloodstream where it is carried by the molecule albumin. So albumin then acts as a bit of an escort to transport bilirubin to the liver. And once it gets to the liver, it undergoes a kind of final step in the traditional journey when you think about bilirubin, which is a process that we call conjugation. Conjugation simply, in this case, implies that it has been made water-soluble by binding it to another molecule called glucuronic acid. So this conjugation of bilirubin with attaching to glucuronic acid allows the bilirubin to now be excreted into the bile and then sent into the intestines. Once it's in the intestines, it's on its way out, getting eliminated in the feces. However, this is where things start to get a little complicated and where most people don't realize there's another journey with bilirubin. It's not done yet. It doesn't just get excreted through the feces. Some of it can get reabsorbed back into the bloodstream, back into circulation in a process called enterohepatic recycling. The process of enterohepatic circulation allows bilirubin and some metabolites that I'll mention in a moment to be reabsorbed from the intestines and transported back to the liver. Now, while it's in the gut, after it's been excreted with the bile that I mentioned earlier, bacteria, so the microbiome, convert bilirubin into urobilinogen. Now, this can be reabsorbed and then excreted in the urine. This is part of what gives urine its distinct yellow color. But it can also further be broken down, if it remains in the intestines, into a molecule called stercobilin, which is in fact a brown pigmented molecule. So this is what gives feces, of course, its distinctive brown color. Now, even still, in this process of metabolizing the bilirubin molecule into these various or alter-colored molecules, it can be reabsorbed and then further used in the body. The recycling process can help maintain bilirubin levels, but it also allows bilirubin to exert some effects beyond just being a waste product. In recent years, scientists have discovered that bilirubin is much more than just some byproduct of metabolism. It has significant systemic effects on metabolic processes and various aspects of cell biology. And this is what I want to touch on. That's what's going to be the heart of this mini lecture today. But before I get into some of those unexpected benefits, I wanted to highlight bilirubin's traditional and historically viewed role, which is negative. And that's appropriate. There are consequences to bilirubin being too high. When bilirubin levels accumulate beyond a certain threshold in the blood, it leads to jaundice, a yellowing of the skin and eyes. Now, of course, depending on the pigment of the skin, the yellowing of the skin might not be as obvious, but everyone's going to have white eyes, and then that is what can become somewhat yellowed in the case of jaundice. This typically occurs when the liver is unable to process and excrete bilirubin efficiently. And that can be the result of liver disease or an obstruction in the bile duct or a problem of the red blood cells where it's not the liver at all. It's just that the body is destroying red blood cells too readily in a process or in a condition called hemolytic anemia. Because jaundice is associated with these types of serious conditions, bilirubin itself has been historically viewed as a toxic product. And it can be. Unconjugated bilirubin, you remember the process of conjugation, that's the process whereby we're getting the bilirubin in a certain state where it's able to be excreted. But when it doesn't undergo that conjugation, it can stay in the blood and then get to the brain and cross the blood-brain barrier and accumulate in neural tissues. This is particularly dangerous in newborns, where high levels of bilirubin can lead to conditions called connectoris, which is a sort of neurotoxic condition leading to some brain damage or hearing loss. So these are serious consequences. Now, that is the historic view, and it is appropriate to highlight it. What I want to focus on now is some of the emerging research suggesting that bilirubin plays a more complex role in human physiology, including some beneficial effects that extend beyond the traditional characterization. Instead of merely being a byproduct of red blood cell breakdown, bilirubin may serve as an important antioxidant and even a metabolic regulator. One of the most exciting discoveries about bilirubin is its potent antioxidant properties. Bilirubin can neutralize reactive oxygen species, or ROS or ROS, which are highly reactive molecules that cause oxidative stress. Oxidative stress is an important driver of a lot of chronic diseases. It does contribute to insulin resistance in a kind of roundabout way. It certainly contributes to cardiovascular disease by enhancing the peroxidation of various molecules and lipids, and even implicated in certain instances of neurodegeneration. But how does it do it? Bilirubin can exert this antioxidant effect by cycling between its oxidized form, biliverdin, and its reduced form, bilirubin. This cycle allows the bilirubin to continuously scavenge free radicals, reducing the overall oxidative damage in tissues, including pancreas and blood vessels and more. Recent evidence has identified that there's also a direct interaction between bilirubin and an enzyme family called NADPH oxidases. This is a family of enzymes that generate ROS, or remember ROS, or reactive oxygen species. By inhibiting the NOx or NOx activity, that's that NADPH oxidase family, by inhibiting the NOx activity, bilirubin can effectively prevent oxidative damage within the cell. Additionally, beyond that mechanism, bilirubin has been shown to activate another molecule called nuclear factor erythroid 2 related factor, so NRF2. This is a transcription factor, and just remember NRF2. That's how it's abbreviated, and it regulates expression of a lot of antioxidant genes. So all of these mechanisms together really illustrate how bilirubin does in fact serve as a crucial defender against oxidative stress. And then now I want to highlight its role in inflammation. But even as I transition from talking about oxidative stress to inflammation, I can't help but wonder as a scientist the main at the role of bilirubin itself. That is there some intelligence? Is there some smart reason why the body may have higher than normal levels of bilirubin when a newborn comes into the world? Might there be higher levels of oxidative stress that need to be reconciled? As we'll get into now, might there be some benefits of an anti-inflammatory effect? So perhaps as much as we look at bilirubin as a problem in the newborn, and it certainly can be, if the levels are higher than what you'd expect, and they commonly are in newborns, perhaps there is some reason for it. Now, as I mentioned, there are some anti-inflammatory effects, and bilirubin has been found to inhibit some key inflammatory pathways, including one of the biggest, which is NF-kappa B. So that's nuclear factor kappa B. That is one of the key transcription factors that controls the expression of multiple pro-inflammatory cytokines, like a lot of the big ones that you've heard me mention or others, like TNF alpha and the interleukin family, for example. By suppressing NF-kappa B activation, bilirubin can reduce the overall inflammatory burden in the body, which is, of course, going to be a potential benefit in metabolic disorders where you may have higher inflammation, like with obesity and hypertrophic adipocytes contributing to a greater inflammation in the body, which, of course, then contributes to insulin resistance. Also, one final point in the inflammation topic is that bilirubin has been shown to enhance some of the function of regulatory T cells. And T cells do, in fact, play a role in suppressing excessive immune responses, all in an effort to try to maintain homeostasis. So, altogether, we have these myriad actions of bilirubin acting at multiple sites to try to control regulation, try to control inflammation, as well as, of course, oxidative stress. Now, I've alluded in a couple times at a couple moments, and both oxidative stress and inflammation together are uniquely relevant to heart disease. And this is an interesting opportunity for me to share with you an interesting disorder, or what's commonly called a disorder, but because of its changes in bilirubin, actually offers a bit of a protective effect. That is a condition called Gilbert Syndrome. So one of the strongest pieces of evidence supporting bilirubin's protective role, again, given its antioxidant and anti-inflammatory effects, comes from studies of Gilbert syndrome. This is a benign, generally considered benign genetic condition in which an individual will have mildly elevated bilirubin levels due to a reduced activity of the glucuronidase. That is the enzyme in the liver that conjugates the bilirubin to the glucuronic acid to help it be excreted. So that enzyme isn't working quite as well. Thus, the bilirubin levels are just a smidge higher than they would be normally, or some instances, decently higher. But again, generally considered benign. These are not people who are suffering from any kind of neurotoxicity of bilirubin crossing the blood-brain barrier. Interestingly, people with Gilbert syndrome have consistently been found to have a much lower risk of dying from heart disease. And there is some speculation that this could be due to both the antioxidant effect and the anti-inflammatory effect of the bilirubin levels. To me, as a scientist, I can't help but note that this condition, Gilbert syndrome, provides a natural experiment, if you will, demonstrating bilirubin's cardioprotective properties. Higher bilirubin levels in these individuals correlate with lower oxidative stress and reduced inflammation throughout the body. And these are both key factors that are thought to play into the development of atherosclerosis and other cardiovascular conditions. Now, a final point that I wanted to discuss is me getting into fat tissue. Now, you understand that I have an interest and even affection for adipose tissue. I want to defend it, and I want to highlight its problems when things go awry. This could be the most unexpected effect, namely that bilirubin has emerged as a key regulator of energy metabolism through its effects on mitochondrial function, particularly in adipose tissue. Studies suggest that bilirubin influences thermogenesis by modulating mitochondrial uncoupling in brown and beige fat cells. Now, as a reminder, brown adipose tissue has its distinctive color, and it is, in fact, a dark reddish brown because it has such a high population of mitochondria. And these mitochondria are not normal. They have this higher degree of uncoupling proteins, which I'll get to more in a moment. But beige fat is sort of this middle ground where you have white fat, which is starting to have more mitochondria in it. And white fat is very white, because it is so loaded with fat. It looks like Crisco or something. And there's so little mitochondria in it that there's no influence on its color. But what if you start increasing the mitochondrial content just a bit? Well, then we don't say that it's brown fat. We say that it's beige fat. Now, what's unique about them, again, both of these, isn't that there are more mitochondria necessarily. It's that the mitochondria are behaving in a bit of a quirky way. Again, that's mitochondrial uncoupling. That refers to the process by which the cells generate heat rather than generating energy through the actions of the mitochondria. And this is facilitated by these uncoupling proteins, particularly UCP or uncoupling protein 1. Essentially, the mitochondria are creating a futile cycle, which is it's breaking down the substance of nutrients, and then normally that is going to be coupled to the production of ATP, the main molecule of energy, if you will, that the cell is going to use to get something done. In this case, however, that breaking down of the nutrient stuff is just used to generate heat. It's been uncoupled from generating ATP. A groundbreaking study published in a very good journal, a classic historic journal, the Journal of Biological Chemistry, or JBC, found that bilirubin supplementation in mice increased the expression of UCP1 in white adipose tissue, effectively converting it into the more thermogenic brown adipose-like state. That is the process of browning or beiging the fat, where you're taking the very kind of sedentary, low energy white fat, enhancing its energy expenditure, which of course is going to contribute, as it did in this study, to some defense against diet-induced obesity and the metabolic dysfunction that accompanies it. This particular study found that bilirubin acts through a master regulator called PPAR gamma. And this is a key regulator that determines the differentiation or the development of adipocytes. In addition to this, related to this, with its effects on UCP1 in mind, recent research has also linked bilirubin to an increased expression of PGC1 alpha. Now, that is an abbreviation for a much longer term, peroxisome proliferator activated, that's the PPA, receptor R gamma. Coactivator 1 alpha, that's the full term, PGC1 alpha. PGC1 alpha is another master regulator of mitochondrial biogenesis and overall metabolism within a cell. A good paper published in Scientific Reports demonstrated that bilirubin-treated rodents exhibited increased PGC1 alpha expression alongside enhanced mitochondrial function, including not only greater fatty acid oxidation, but also just overall energy expenditure. So altogether, this suggests that bilirubin not only promotes thermogenesis, which is the uncoupling where the mitochondria are just burning nutrients for heat, but it also supports overall mitochondrial health or fitness, if you will, and a generally favorable function at a broader level. Again, with a large, heavy focus on the mitochondria within fat tissue. In human studies, this is a little more unclear, but individuals with Gilbert syndrome, those are the ones who naturally have higher bilirubin levels, tend to exhibit a lower body mass and generally appear to be somewhat better off when it comes to insulin sensitivity. This could be a result of the enhanced mitochondrial function and the uncoupling leading to an increased basal metabolic rate. Now, this still needs to be really clarified, but if these ideas apply to the whole human, it would explain how someone with Gilbert syndrome would be somewhat protected from metabolic disorders. But all of this is interesting enough that bilirubin is being explored as a potential therapeutic target. Where there's such a never-ending pursuit of therapies and drugs and interventions to try to help solve for obesity and insulin resistance. Now, this is kind of being added to it. Interestingly, along these same lines, bilirubin has been linked to metabolic flexibility, the body's ability to efficiently switch between fuel sources like sugar burning and fat burning. And of course, that's largely a function of just improved insulin sensitivity, because it's insulin that largely determines the burning of the fuel. But there's also that relevance of enhancing mitochondrial function and biogenesis by promoting PGC1 alpha. By activating PGC1 alpha, bilirubin can just put the cell or the body by extension in a state to help it bounce between those two. Because remember, metabolic inflexibility is when the body is generally stuck in sugar burning mode. Well, that's a state that can occur outside of the mitochondria. So if you have more mitochondria, it just makes it more likely that you can more easily be in fat burning, which of course requires a greater reliance on mitochondria. But given its broad mitochondrial effects with thermogenesis and biogenesis, etc., bilirubin does represent a pretty interesting, exciting area for obesity research. The potential for it to improve these metabolic outcomes absolutely suggests or points the finger at bilirubin. Now, there are some lifestyle changes that a person can make. You may be thinking, well, I want to increase my bilirubin. One way, although this isn't clear, one way that there's a little bit of evidence to suggest you may do this is remember how bilirubin comes out. How do you release it? Because for this all to happen, the bilirubin is getting dumped into the liver and then it's getting reabsorbed into the intestines. And remember how it's getting dumped, well, from the liver into the intestines is what I should have said. So how does it go from the liver or to the guts? It does so through coming out with bile. Well, when are you releasing more bile? When you're eating fat. And so if this is something that you are thinking I want to try to take advantage of, it could be that by following generally a low carbohydrate diet that's going to be higher in fat, you are going to be releasing more bile. There's no doubt about that, which is going to have benefits on the gallbladder and help preventing gallstone formation. That's very, very well known, but it's also a tangent. But as you're producing and releasing more bile, you could be benefiting by enhancing this enterohepatic recycling that I started with, where the bilirubin's coming out into the guts and then getting reabsorbed. And it's as it gets reabsorbed that you have the first time that the bilirubin is now really acting in a systemic circulation way. The natural kind of experiment, if you will, that I mentioned earlier of Gilbert syndrome does reinforce some of bilirubin's protective effects. So bilirubin is not something to fear. Like most things, if you will, the dose makes the poison, that if you have too much bilirubin, it can start to compromise brain function. But slight elevations could be beneficial. But suffice it to say, my hope in giving this mini lecture is that by understanding some of the full range of bilirubin's effects, that not only are you more familiar with this, but it could be just helping you understand some of the new avenues that are there and being pursued to help in the, well, never-ending chasing down of some therapeutic strategies, just to give anyone whatever advantage they can get. Thanks for tuning in. I hope that you found this lecture insightful and that you feel like you now have a much better idea of just what bilirubin is, why it's sometimes a villain, and why in some instances it might in fact be a bit of a hero. Until next time, more knowledge, better health.